So, aseptically, what does that mean? Without contaminating. Because we already know from our very first lab, is there microorganisms in the air?
Yes.
Yeah. On our desktop?
Yes.
Our hands?
Yes.
Breathing?
Yes.
Yeah, right? So we want to minimize that. We don't want to breathe on our cultures. We don't want air falling into our cultures. We don't want stuff from our desktop getting into our cultures, right? So we're going to use a particular method that's going to minimize the chance of contamination to as minimal as possible. So aseptic is, again, that describes, it's an adjective, describes the condition of being without contamination. The other thing to take into consideration, too, is we don't want to contaminate our environment. We don't want to contaminate our cells, our desktop. If we do, we're going to disinfect. I thought I brought my bottle up here. Probably, didn't make it. Anyone else see the other Lysol? Yep, I did make it. Once it burned blood. So this is Lysol, right? We have 2 spray containers that I bought with my own money. So I get tired of us pouring it. I wanted to be able to spray it. That we'll use to disinfect our countertops, our desktops before we start and when we get done. And definitely if we have a spill, we're going to disinfect, right? So mixed culture, as we've already said, means that it's 2 or more organisms. A pure culture, hopefully you wrote that down today, right, on the quiz. It was the bonus. Means what? 1 species. 1 species. 1 organism. That's it. A lot of times that's what we want to work with, right? Because we want to know our results are from that 1 organism, not is it this one or that one or whoever happens to be in there. So times we start out with a mixed culture like what you guys have today, right? We purposely mixed 2 organisms in that tube for you today, right? You need to be able to separate those out to be able to study them individually as pure cultures. The rest of the cultures, it has 1 name on it, right? So unless it's been contaminated, it should be that single organism in those tubes on that plate. So there's different isolation techniques, but the whole point is if you have a mixed culture of more than 1 organism, you want to separate them out so that you have individual organisms. One of the most commonly used techniques is the streak plate method, which is the one we're going to teach you today. And this utilizes a plate. So we have a large surface area. And typically we're going to use this method when you have what's referred to as a heavy inoculum. It means your culture has a lot of organisms. So if you guys have noticed, your mixed culture is very cloudy, right? There's lots of bacteria in there. This is going to be the best method for that type of starting sample. Colonies are growths you see on solid culture media, so on the plate or in that slant in the tube. We use a lot of slants in this lab because it creates a solid surface within a tube, and the tubes are much easier to store a whole bunch in racks, in the incubators and refrigerators, than keeping track of a whole bunch of plates. But for some techniques, you need that large surface area like the streak plate, so we will use a plate. And we will use some plates throughout the semester. We had some blood agar plates, but I forgot to refrigerate it. And you could see, the differences based on that different type of food. And that's what the media is, right? It's food for the microorganisms. So on those solid culture media, they'll form these characteristic colonies, these growths that you'll see. Now, if that growth is from a single cell or a cell from the same species, we refer to those as colony-forming units, abbreviated CFU. And later on, we'll even count these. And the reason why we say colony-forming units as opposed to saying, oh, it came from a single cell, as you guys will find out next week, or next week for you guys, when we start staining and look at these, some of these cells stay next to each other when they divide and they cluster, and so you can't really get a single cell by itself. But the good news is they're all the same species, right? That's why they're stuck together. So aseptic transfers, again, transferring where contamination doesn't occur. So you start with a pure culture, a single species, hopefully. And it could be in any of these forms, right? It could be on a plate, on the solid culture media in a petri dish. Could be in a tube, in a slant orientation. Could be in a broth, a liquid food source. And then you're going to transfer them to sterile media, media that is devoid of any microorganisms. And, you could transfer a broth to a broth, where you're taking organisms from a broth culture and putting them into another broth. You can go to slant, or you could go from a plate to a broth, like we're going to do today, right? Inoculation is the introduction of the bacteria into that sterile media, right? So you're taking some bacteria and you're inoculating a sterile media source so that it'll grow. Inoculum is what you're starting with, right? Those organisms that you're taking from whatever source, that's your inoculum. Now you could pick up a lot and have a heavy inoculum. That's typically not what we want to do, because microorganisms are just that, microscopic. You only need a small amount usually to be able to grow up a whole bunch more. So we want to develop the technique of doing a light inoculum, taking a small amount. Aseptic transfers are sometimes performed using sterile technique. This is sometimes interchangeable. For me, it kind of doesn't make sense, right? Because sterile means what? No microorganisms. If you have growth, it's not sterile, right? Seems counterproductive. And some of the techniques, too, that we'll use in this lab aren't necessarily always employed. It depends on the circumstances. So if next door was open, did Peter still there? Yeah, maybe we can pop over there in a second. Before we start performing, if he's still there. They have what's called a laminar flow hood. So this has special filters on it that filter the air and circulate the air that creates this curtain of air. So the inside that hood is a sterile environment. So you don't have to do some of the steps that we're going to do because the environment itself, right, is clean. Unlike for us, we've got microorganisms in the air, right? So we have to constantly keep flaming things, even the air within the opening of our tube. And that's one of the ones that I tend to skip, right? Is that the one I skipped in the video, right? I didn't flame the mouth of the tube. And again, because it wasn't something that I always had to do, right? I worked in a different environment most of the time when I was working in research than what most microbiologists do. I did a lot more cell culture, a lot less bacteriology, a lot less working with bacteria. Make sense? Okay. Huh? Yeah. So, so even me, 20 some odd years of experience, I still have to, remind myself certain steps, right? So, you guys are going to be my lab partner. I'm going to actually have to think today and purposely make a mistake in my demonstration. And you guys as my lab partners, I want you guys to point it out to me, right? So I'm going to get you guys in that practice mode, right, of helping each other. So food. What kind of food do we give them, right? So we use a lot of general purpose media in this lab. So beef extract. So they like hamburgers just like we do, y'all, right? It's a very rich food source. It's got just about everything you need. Protein, sugar, fats, you name it, right? They're animal cells from a cow in powdered form. Peptone is a digestion of protein. So you'll see here, enzymatic digestion of animal protein is what peptones are, right? So instead of being that whole big protein, they've digested it some. So basically we're even giving them baby food, right? We've pre-digested it for them. So just like how we grind up baby food for babies because they don't have teeth or don't have many teeth, right? They'd be sucking on a carrot all day if you didn't grind it up for them, right? So we kind of grind it up for them so they'll eat faster and grow faster. Trypticase soy agar is another one we commonly use. And again, the agar is the polysaccharide derived from marine algae. It solidifies the media. So when it cools, after it's been boiled, it's going to create that solid surface, whether it be in the plate or in the tube that's been slanted when it's cooling. So tryptone is an enzymatic digestion of casein, which is the white protein found in milk, right? So it makes your milk white. Trypsin is a pancreatic enzyme commonly used to digest that protein source. And then we also digest soybeans. That's the soytone. And then some salt for isotonic balance, right? Sodium chloride. So these are all rich, right? Lots of nutrients there. Abundant source of nutrients, but not well defined, right? We don't know the exact composition, right, of what is there. We don't know exactly how much sugar, how much protein, how much fat. But we don't need that, right? We need them to just eat and grow, right? So that we can play with them. Rich media, again, typically just used for routine growth, propagation of the microorganisms. But we can get more defined, right? Some media I equate to the Nutrisystem diet. We know exactly how much everything is in there. That's expensive though, right? Nutrisystem's not cheap, right? We only have one media that falls into this classification, and the only reason is we're using it for specialized testing. And so therefore it happens to be all laid out exactly what's in it. It's all measured out. We know exactly how much of each thing is in it. Right? There's no ambiguity from batch to batch like you'll get with nutrient agar and trypticase soy. Selective medias go one step further. They help us select for particular organisms, right? Based on what's in that media. We actually, that blood agar plate that I used, right, had antibiotics in it that select for gram positives because it kills off the gram negatives. There was also an enriched media. It contained whole red blood cells in it. So it helps us differentiate organisms that can destroy red blood cells, whole red blood cells, from ones that cannot. So the second half of the semester, you're going to be given an unknown bacteria. And you're going to employ some of the techniques you're going to learn this first half, mainly staining. Right? So we're going to give you that organism and you're going to stain it and look at it under the microscope based on the different stains, and then we're going to start using different media, selective and differential media, to learn characteristics about your organism. You're going to be able to figure out what you were given. So why do we transfer bacterial cultures from one media to another? We want to propagate them, to revive the stock or make a culture that's actively growing. When we stick them in these tubes or on these petri dishes, it's like sticking them in a fallout shelter with a whole bunch of MREs and the toilet just broke. Not a cool environment, right? Eventually they're going to run out of food. Worst case scenario, though, they're going to run out of places to put their waste, and it's going to become toxic. So they're going to stop growing. They may even start dying, right? They're not having any more babies in that environment, right? That's not where you would want to be having some babies. Right? So they stop growing, right? They even start dying. So you got to rescue them, right? And guess what we do? We pick them up out of that fallout shelter and we stick them in another one. Right? And we just keep doing that over and over and over again. So we use a loop, right? So I got some nice new loops for us to use. If it's getting bent, you're applying too much pressure, right? You need a lighter touch. And you definitely want to straighten it out before you heat it in the flame. Right? So you want it nice and straight. The loop helps us grab the microorganisms, and I'll show you how to use that. Sometimes we need a very small sample, or we have to do what's called a stab. And in that case, we need a needle, right? So as you can see, it's just a straight wire. Today we're going to use our loops. And I got some more that are kind of cruddy, and I need to Remember to bring my wire cutters in and make some new loops, because I got too much on them. And that's the thing, is if you pick up too much organism over and over time, the minerals and the ash will stick to the loop and it'll gunk it up, which makes it harder to get it clean, and therefore you contaminate your cultures, right? Because you're not actually getting them all off that loop because it's encrusted in the little nooks and crannies on those loops. So differential is selective, right? We use this to identify the organisms and the different properties they have. We'll use a loop, but sometimes you may require, again, a needle for that kind of inoculation. Differential, it's a wide range of organisms will grow, but when they do grow, they're going to change based on the characteristics of that media in that organism. So in the case of the blood agar plate, those organisms actually poke holes in the red blood cells to get at the hemoglobin inside. And more so, they're after the iron component of your hemoglobin, because they need iron just like we need iron. They need it for their metabolism as well. So that's why they attack our red blood cells if they can. It's a very rich source of iron. So other things that we see are fermentation of sugars, right? We can detect this using specialized media. Selective media grow a narrow range. In this case, we're selecting, right? We only want a particular group of organisms to grow. In the case of the blood agar plates, it contained antibiotics that would kill off gram-negatives because we just want gram-positives to grow. Or another one we use has a really high salt content, so only ones that can survive in that very salty environment grow. So why is it important to use sterile technique? We want to keep our experiment reliable, right? Keep out in the environment from our samples. We don't want stuff from the air. We don't want stuff from you breathing, or from your fingers, or from the desktop. So that brings me to the quiz from last week, right? So that Varnado story, I didn't make that up. That really happened, right? So they put Petri dishes in the restroom. Dr. LeBlanc walked in, right? There was a male student coughing all over it, laughing. You know what she did, by the way, what I left out in that story? She doesn't have children. She put a sign, "Please do not touch." What does that say to a little kid? Touch me. Like whenever you tell them don't, why? Right? So what happened to their experiment because that guy went over and went all over their Petri dish? It's compromised. It's contaminated. But what does that mean? Can they use the results from that dish? No, they got to pitch it and start all over again, right? They're not testing air quality anymore. That dude purposely contaminated their experiment. That's what I was looking for, y'all. To understand that that experiment was null and void. Right? They were no longer testing air quality. They were going to have to repeat it. I got some really interesting answers. I gave partial credit, right? For those of you guys who are kind of on the right path. But that was the important kernel that I was looking for. That experiment was null and void. Right? They had to start over. Make sense? So, we want our experiment to be reliable, right? We don't want someone coughing all over our Petri dish. We want to keep our original culture pure. Just the organism that we're studying, right? Just that one. Make sure that our sterile media does not become contaminated with one or more additional species, right? Again, we only want one. So then we know the results are for that one organism. We don't have to go, well, was that this organism or that organism that made it turn blue? Who knows, right? If you have more than one. You don't. Safety. We want to keep your sample out of the environment, right? So again, if we spill, we want to disinfect, right? We don't want to put our caps to our things down on our bench because it might have microorganisms on it that's going to then contaminate our bench, right? If we don't clean up, then it's going to get on our stuff and we're going to take it out of this room, right? Or someone else is going to come in and get exposed to it. So make sure our possessions, right, we put them away, right, so that we don't get unwanted organisms on stuff. Make sure no one else in the laboratory becomes contaminated with potential harmful microorganisms. Klebsiella pneumoniae can cause a really serious form of pneumonia. No licking Petri dishes, right? Nobody wants this, right? You'd have to come in contact with large numbers of these organisms to get sick, in direct contact with the Klebsiella pneumoniae, right? But we don't want to contaminate our environment, right, where they could potentially grow to large numbers and someone accidentally get exposed to these things. So having said that, when we get done working with our microorganisms, right, we're going to take our goggles off, we're going to put them away, we're going to take our aprons and stuff off, we're going to take our gloves off, those can go in the regular trash, and we're going to wash our hands, right, before we leave this room. At some point you have to get up and go to the bathroom, you got to do that procedure, right? We don't take this stuff out of the lab because it could be contaminated. Make sense? Okay. So if we want to isolate a single species, so say we have a broth that contains more than one, a slant culture or a plate culture contains 2 or more, we can put them onto sterile media, say a plate, and we can do it in a method called the streak plate where we're gradually spreading out the organisms. So hopefully we're separating them into individual cells or clusters of the same species so that they'll grow up into visible growths that we can see on a plate known as a colony, right? Or a colony forming unit, if hopefully it arose, right, from a single cell or a single species. And then in that case it may even be a pure culture. Here you can definitely tell they started with a mix, right? You see different colors. That's a pretty good indication you may have different organisms there. But some organisms will grow differently depending on temperature. So I always know when we lose electricity if I'm growing one of the organisms, because at 37 it's white, at 25 at room temperature it's red. So if I had polka dotted growth once on my Petri dishes because it started out at 37, right? And then we lost power over the weekend and it was still cool out so it didn't get really hot in here and it changed color, right? So if the inoculum is light, you can maybe do a single streak, and that's what we did, right? There wasn't a ton of bacteria on our desktop, so we were able to take a sample and just do a single streak, right? Just rub that swab down the plate, and we got a bunch of different colonies, didn't we? It wasn't completely flooded in growth. Same thing with the leaf sample if you looked at it over there, right? We got individual growths and stuff spread out. They started to grow together over time, right? But there wasn't a large sample. Today we're starting with a large sample. So this quadrant streak method of having several sections, right, spreading them out is going to help us isolate them out, right? Isolating meaning you have a colony that's not touching others, right? It's all by itself, right? So hopefully, again, the hope is it's pure. Hopefully, it's a single species where we started with more than one. So there's several different ways. A lot of times, beginning students, all right, when we're first learning, we draw ourselves like kind of a little road map on the bottom of our plate. So I'll show you how to do that. This one, there's kind of angled lines. I'm not that high tech, right? But this is our plan, right? We'll start in 1. We'll move organisms from 1 to 2 to 3 to 4, right? So we'll take the organisms from the sample, and we're going to do what I call invisible coloring, right? We're going to color in. We're going to move our loop gently across the surface and try to color invisibly, right? You're not going to see anything. That entire section with organism. Then we're going to flame our loop. That removes all the organisms, right? Turns them into ash and minerals. Completely evaporates all the water. We're not going to go back into our sample. Instead, now, we're going to use our loop to pick up some of those ones we already put on the plate and spread them into the next section. So we're basically diluting as we go, right? We're picking up less and less organisms. So again, we're going to try and cover as much surface area as possible. Notice we're going to go and pick some up, right? And then we're going to spread them. And we're even going into the next quadrant so that we can then pick them up a few times to pick some up and then spread them out in this third quadrant. And then we're going to do it one more time into the fourth. That's one technique you could utilize. Each time, in between each quadrant, we flame our loop to remove the organisms from the loop. And so we're only picking up a little bit from each section. You're always going to work with a loop, but sometimes your beginning sample, you may actually use a swab and then a loop in that first section, depending on the circumstances. Because, like, think about this. If you're doing a patient sample, right, they're not sticking a loop up someone's nose or rubbing it into their wound, right? They're using a swab, and they're going to pick up a sample. You're going to get a swab at the lab. You're going to take that swab and rub it on a small section of the plate. Then you're going to use your loop to streak it out. So I like the straight lines, like a goal post. I can handle that. Right? Same thing, right? We're going to spread it. This one, they omit some of the lines, right? This is a more high-tech. I don't even draw lines anymore, y'all, right? I can keep track of where I'm on the plate and where I'm going, right? So notice that one that went around with the yellow growth. You don't see any lines. I didn't erase them. There weren't any lines to begin with, right? But I still managed to do that pattern around the plate. So again, fill up one, grab to the next. You know, it's just helping you keep track of where you're going. Now, this is a technique that I typically do for the last quadrant, is I don't do a continuous streak. I do several streaks. So I grab my loop and I drag across and I pick it up and I drag across and I drag across the whole surface here. You can do either one. Just what do you need to do? Write down which one you did. Document. So you know when you get your results, right, what each one would look like and what you did. With practice, lines don't have to be drawn anymore, right? When you've done as many as I do, you don't bother drawing the lines anymore. You know what you need to do. Okay. So Peter Hickman, that was bopping in and out of here. Is he still over there? Yeah. Worked in a lab where he had to do a lot of samples. So can you imagine having to do 8 Petri dishes? So instead, he would actually divide a Petri dish into 8 sections and do tiny little streaks in each section. So he'd use sterile toothpicks, right? And he'd use the end of each of the sterile toothpicks. So from his sample, he would do one little line and then cross that little line, right? Flipping the toothpick, flip the toothpick again, and then one more time, and then streak that down. That would be one of his samples. Then he'd move on to the next one. He could fit 8 samples on one plate. Right? So growth. Growth is going to look different based on the media, right? Here you can see it growing on the surface, but in these broth cultures, this one's kind of cloudy all throughout. It might be a motile organism. It's swimming around. These guys are just at the top, right? They're probably aerobic, and if the culture isn't shaken or stirred throughout incubation, then this is the only place they're going to get oxygen, so they're going to stay at the top of that tube. So we had a couple, and there's a test we'll do later too, that do that. They're strict aerobes. They're going to stay right at the top where the oxygen is. We don't use broth very often because of that problem, that oxygen doesn't diffuse real well through it. So especially aerobic organisms, they're not going to be really happy because they're kind of stuck up at the top. And the research lab that I worked in, we had this wheel, almost like a Ferris wheel, in the incubator, and had little holes in it. We stuck the tubes in, and it spun them during incubation. But we don't have that, so we use mostly solid culture media in the tubes, again, to save space. So you can see these guys are kind of whitish, right? This one looks how? It's kind of chunky looking, huh? Whitish, kind of chunky looking. And this one, y'all, believe it or not, there's bacteria on there. They're translucent. So you might get an organism, or we might work with an organism and be like, Miss Acker, you gave me an empty tube. I said, no, look closely. Right? They're translucent. It's kind of hard to see them, but there's growth on that slant. Trust me. There are bacteria there. All right, so hopefully you've wrote some descriptions. And we're not going to go to this level, but especially on solid media on a plate, you can kind of see the different morphologies to the elevation of the colony off the plate. And then the margin, like for instance, if you look at the Klebsiella, it kind of looks irregular, right? It's got bumpy edges. We're not going to learn that terminology or necessarily utilize that terminology to that extent. But I definitely want you guys to notice if stuff is smooth, if it's dry looking, any color associated with it, if it has rough edges or smooth edges. So in this case, this is Staph epididymis, and you can see again white growth. This is on a blood agar plate. If you were to be able to look at it, it'd be raised. Notice they're circular, the colonies, and they're relatively smooth. Klebsiella pneumoniae is kind of a whitish color as well. It's raised. See how it kind of looks like a little hill? It's circular for the most part, right? Mucoid, sometimes it even looks mucousy, right? Really shiny. Micrococcus luteus, this one is pretty easy to identify because it's yellow, but it's not the only yellow organism out there, right? But that one definitely has unique color pigmentation to it. It's kind of pretty. And again, notice how it's kind of like a little hill when it grows, right? Again, circular colonies, they're smooth. This is a different bacillus than what we're using today. This is Bacillus subtilis, and it's white. It's raised, but look at even how time affects it, which you can definitely see with our Klebsiella, right? Those were done Wednesday, so they're over a week old, right? So some of these organisms are screaming to get out of the fallout shelter right now, right? They're like, please rescue me. But you see how it's irregular, right? And you have these different layers even forming here. So, use your good scientific skill of observation and recording and observing these things. Each time, I give you an organism, look at it, right? Is it white? Is it creamy? What does it look like, right? And write it down. So again, this is the plan for today, right? Which actually it's kind of written out on our labels on our tubes, right? So I'm just going to follow that because I'm going to turn off this so that you guys can come up front and watch me actually do the transfers. Then you're going to go back to your lab bench, and as I said, you're going to work in pairs next to each other. One person will do the aseptic transfers while the other one watches, and then you'll switch. The other side of the lab bench will use the mixed culture and do their streak plates, and then you guys will switch. Okay? Let me turn off this light. Oh, let me hit